Hey BioTeam. So there's a couple of things that all cells have in common. At their basics, cells are made up of membranes and ribosomes. So membranes we've already talked about a little bit. Those are flexible barriers that keep the insides of the cell inside, keep the outsides of the cell out. And if we were to zoom in on it, we'd see that they are made mostly of phospholipids with this charged phosphate head and these lipid tails that form kind of this double bilayer, two layers of phospholipids. But you'll see that there's also some protein channels and gates that are embedded within that phospholipid bilayer. And it's for this reason that we say that membranes are semi-permeable. That is, they have the ability to let some things permeate through them. Ribosomes, meanwhile, are responsible for building the proteins that make up the cells. And what's interesting is that all cells, whether they're bacteria cells, animal cells, plant cells, all cells have ribosomes that work and function almost identically. And so they provide evidence that all living things are related, that there is a common ancestor that all living things descended from. And in fact, these ribosomes are so similar to each other that if you were to take the nucleic acid from an organism like a human, take the DNA from a human, and insert it into a bacteria cell, the ribosomes in the bacteria cell could use that DNA to build proteins from it. So this is all cells at their basics, membranes and ribosomes. But some cells, eukaryotic cells, have more complicated internal membranes inside of them. So they have the big membrane around the outside, but smaller membranes inside. So for instance, they have nuclei, where nucleic acids are stored. And they have endoplasmic reticulum. So they have rough ER, which is this labyrinth-like, maze-like membrane with ribosomes inside of it, which provide these rooms and compartments for the building of proteins. And they'll have smooth endoplasmic reticulum, or smooth ER, which can provide the rooms and compartments for building lipids and for detoxifying the cell. They'll have Golgi bodies that can fold and modify proteins after they have been synthesized, and then package them up for shipment or secretion inside of tiny membranes called vesicles. Another type of internal membrane would be a vacuole, which is a storage membrane. It can store food or waste or water. And in plants, actually, this water vacuole can get so big that it creates something called turgor pressure that supports the cell, prevents it from collapsing. Another type of internal membrane would be a lysosome, which would be a membrane that contains hydrolytic enzymes, or enzymes that can do hydrolysis, that are able to break down food and waste products inside of the cell. Chloroplasts, which are membrane-bound organelles that can do photosynthesis, and mitochondria, which are able to do aerobic respiration. And one thing worth pointing out here for mitochondria, and I guess chloroplasts too, is that mitochondria have something called a double membrane. So they actually have this smooth membrane around the outside. You can see that on the chloroplasts here. But then on the inside, there's this more complicated, wrinkled-looking, folded and creased membrane inside of it. And so you'll notice that actually a lot of these membranes, the Golgi complex, the endoplasmic reticulum, have these crenellations. They have these folds inside of them. And the reasoning behind that is that having the folds increases the membrane's surface area. And having a really high amount of surface area, having a lot of space at the surface of a membrane, allows materials inside of it and outside of it to enter and exit quickly. And that's it. At this point, you guys have some practice problems. Best of luck.